good morning or good afternoon and good evening, depending on where the world you are at. Thank you for your patience with a little bit of technical difficulties. The first time we've experienced it, but I know other people have run across these with Symposia in the past. So again, appreciate everybody's patience as we kind of got things worked through. Now I'll turn my camera on just to be good and sociable to see each other once again. But welcome to the AGSx Symposium Spring 2022, session number 3. We're at the Arthropod Genomics and Genome Engineering, hosted by or moderated by Dr. Lindsay Perkin. So next slide, please. So this is the
easier said than done. It should be going.
It might be a bit of a lag, but just want to obviously this is the 3rd session in our series. We have one more coming up on May 10th, moderated by Marcy Lorenzen. A little bit of help. We're getting everything is on the website. We'll cover that in the next slide, so you can go there and check out all the information about this and the upcoming session 4. So next slide, please. So, and of course, like everything, this is not done just by a few people, but this is a really great and interactive team, but very productive on getting things kind of worked up in the background. Everything for running the Zoom meetings. Thank you, Glenn, for doing that. Anna, who put together the website and handles all the Twitter feeds for us. Pia Olson, who does the video recordings. Well, we have Amanda Stalkey stepping in this morning to help us with the playing of the recorded talks. Rob Waterhouse, who has set up and administrates the Slack channel, and will be giving additional information on that as we get going. And of course, our administrative support from USDA has made all this possible for all the, I guess, the technical background and the facilities to do so. So next slide, please. So again, just going into the website where you can find all the information is we put into the chat, so you don't need to try to write all this information down. And the registration link, just in case you have not registered for session number 3, is located here and also will be posted into the chat as I get done with this. And all the videos for all the AGSx sessions are on YouTube in the I5K YouTube channel. And our Slack channel link is given here. But if you are registering for the very first time, we need to give you a registration link to utilize. So be patient, we'll put that into the chat as well for all the information you need. So next slide, please. And want to give you a very quick plug for the Arthropod Genomics Symposium, which is a hybrid in-person and virtual event that's going on June 9th to the 11th. And there are these registration links right here, and there's a couple upcoming deadlines with that. So if you're not aware of that, please consider attending. It's been always very useful and very informative in the past, and is obviously the parent event of the AGSx. So definitely want to see that keep going on successfully and vibrantly. So next slide, please. And here I'll turn it over to Lindsay. So thank you, everyone.
All right. Hello, everyone, and welcome. And again, we apologize for all those technical issues. This session is the Arthropod Genomics and Genome Engineering session. It was named originally for a PAG workshop, and it's kind of evolved into more than just a session to describe genomes. And to me, and kind of encompassing all the speakers today, the purpose of this session is not only to describe genomes, but to discuss how we can use these genomes to answer biological questions and solve problems in arthropod pest systems. Today we have 3 wonderful speakers. The 3rd talk's actually a hybrid, but I believe only Perot will be speaking in that session. As these talks are being presented, please use the Q&A if you would like to ask. And at the end of the session, we'll do live question and answers. Please don't put it in the chat. I may not see it if you put it in the chat. So with that, we'll go ahead and start with our first speaker. Oh, and I also want to mention, because of us running late today, if you cannot watch the entire symposium, fear not, because these are recorded and they will be posted on the I5K YouTube channel. So you can always go back and watch them if you have to rush out at the end today. All right, so our first speaker is Noah Rose, and he is a postdoc in Lindy McBride's group at Princeton University. And he's going to be talking to us today about the origins and spread of preference for humans and mosquitoes. So I will stop sharing and I will let Amanda cue up his talk.
Sorry, let me make sure that I get the audio just right.
Should be share sound in the lower left. Share computer sound.
All right, hi. I'm excited to be talking to you all today about mosquito evolution and the emergence of mosquito-borne disease in the human era. Let me just get this set up. Okay, so I just want to start off this talk by actually, before I show you any pictures about mosquitoes or anything like that, just by having everyone take a look around and, look where we are, look how the format is. We're in this sort of funny virtual format and we're all sort of separate. It's because of the sort of ongoing, still playing out COVID-19 crisis, where the emergence of this novel virus has led to, a lot of major social sort of consequences. Now, COVID-19 wasn't actually the first time we ever saw anything like this, and another recent example of the emergence of a virus that was, a major disruption was the emergence of Zika, and so. We're going to go back a couple of years and remember when Zika was causing all sorts of problems, including these terrible cases of microcephaly that were documented, especially in South America. And so when it comes to the emergence of a virus like this, what are the actual factors that lead to the emergence of mosquito-borne diseases like Zika? And also, how do evolutionary and ecological forces shape the emergence and spread of disease in general on a changing planet? Well, in the case of a virus like Zika, it might be circulating in animal populations spread by an animal biting mosquito in a forest environment. And you might have occasional spillover of a virus into rural human populations by a vector that bites both animals and humans. And then eventually, you might get a virus spilling over into urban populations where it can spread quickly and cause major urban epidemics. But for that to happen, you need a mosquito vector that lives in human habitats, that likes to bite humans, and that's capable of transmitting the virus in question. Now, a really great example of a vector that fits that bill is Aedes aegypti, which is an aggressively invasive urban specialist on human hosts, and it's the primary vector of yellow fever, dengue, Zika, and chikungunya. And I can show you just how much Aedes aegypti loves humans using this assay called a two-port olfactometer assay, where you put a bunch of mosquitoes in a big plastic box, and you let them decide, are they going to try to go into one trap that leads to a human host, try to bite that human. There's a piece of screen in the way, so they don't actually get to bite them, but you see that they're trying. Or are they going to try to bite a non-human host? In this case, in the illustration, it's a guinea pig. And so here's a video of a olfactometer trial looking at an invasive population of Aedes aegypti from Florida. Now, given the choice, you'll see this mosquito chooses human, another one chooses human, another one chooses human. We can speed up the video a little and see what happens with a larger number of individuals. They have a really strong and robust preference for human hosts. Every once in a while, one will pick the non-human host. So I think you'll see one coming down the guinea pig tube any moment now. There it is. But it's a really strong preference for humans. Now, these Aedes aegypti wasn't always the globally, the super successful vector species across the global tropics that it is today. In fact, it's thought to have spread from its native range in sub-Saharan Africa to the Americas in association with the transatlantic slave trade about 500 years ago. Soon afterward, there were the first major outbreaks of yellow fever. It's then thought to have spread back to the Mediterranean, where there were outbreaks of yellow fever and dengue, although these populations of Aedes aegypti have since gone extinct, but not before they spread with the opening of the Suez Canal to South and Southeast Asia, where there were major outbreaks of dengue and chikungunya soon afterward. course, much more recently, we had the emergence of Zika on Yap. This was initially vectored by a different mosquito, Aedes hensley, but subsequent outbreaks were of course vectored by Aedes aegypti, especially in South America. Now, across its native range, there was, up until I started the work I'm going to tell you about, it wasn't totally clear what was going on with Aedes aegypti. So in its invasive range, it's this amazing, super human-preferring vector. But in the native range, it wasn't sure. We weren't sure if all populations were quite as specialized than humans. In part because early vector biologists had actually gone to some forests in East Africa and Kenya, and they were able to find populations of Aedes aegypti that were living in the forest, biting animals, not really posing much of a danger to humans. They were breeding in tree holes and rock pools. They have this very sort of Edenic natural lifestyle. And so the question that I had at the beginning of my postdoc was, how did this evolutionary transition happen? In particular, what does contemporary diversity look like in Aedes aegypti in its native range, and what ecological factors drive specialization on human hosts? What are the genomic and historical origins of this shift? And what does this what does this tell us about the changing relationship between humans and nature? So in the first part of my talk, I'm going to talk about the ecology of specialization on human hosts. And to start to answer some of these questions, we went back and collected Aedes aegypti eggs from a variety of different habitats across its native range in sub-Saharan Africa. These range from sort of scrubby secondary gallery forests and patch forests where the ancestral animal-biting form of Aedes aegypti really does well. So this is the kind of habitat where you find a lot of animal-biting Aedes aegypti. We found a lot of them here in southern Senegal. But we also collected them from habitats where they were exposed to both human and non-human hosts in sort of natural and more human habitats, ranging into more and more human-dominated areas. And these collections ranged all the way to the northern limits of Aedes aegypti's range in sub-Saharan Africa, in the Sahel region, where there's not a lot of that natural forest habitat, but it does really, really well living in close association with humans, especially in big cities like Dakar. Now, I want to acknowledge off the bat that this work was super collaborative, and it would not have worked if it wasn't for major contributions of Joelna Corley, Diego Ayala, Atanas Badolo, Massam Basila, Ogie Arribador, Joel Lutomia, my fantastic thesis student Nana Ibe, and Samson Otu. Now, these collections were carried out across two major sort of axes of ecological variation. So first of all, they were carried out across very different levels of urbanization. So some collections took place in places like Kumasi, Ghana, where there's over 2,000 people per square kilometer. And you can see in the satellite photo that the landscape is totally dominated by human structures. On the other end of the spectrum, there's places like Bantata, Senegal, where there's only 12 people per square kilometer. It's really like a very natural habitat for Aedes aegypti, not a lot of human hosts, not a lot of human habitat. The other major axis of ecological variation had to do with the climate. So in some places, like Ngoye, Senegal, this is in that northern edge of Aedes aegypti's range. It's a highly seasonal habitat where there's a long, hot, dry season and then a short, really intense rainy season when mosquito populations blow up. And then on the other end of the spectrum, there's places like Kakamega, Kenya, where it's really aseasonal. It's muggy and rainy all year round, and there's always good mosquito habitat. There's lots of standing water, basically. From each of these locations, we established 2 replicate laboratory colonies from egg collections wherever possible, and we selected individuals from different egg traps to get an unrelated sample of individuals for population genomic sequencing. And across this effort, we got 50 mosquito colonies from 27 different locations and 345 unrelated individual genomes. Now I'm going to just jump right into the behavioral data and show you what we got. So on this plot, you see the behavioral preference of different mosquito populations for humans across Aedes aegypti's native range. And each circle here represents a single population. The size of the circle represents the human population density, so bigger circles are bigger, denser cities. And then the color represents preference for human hosts. And so redder circles show a stronger preference for humans. And you can see there's quite a bit of behavioral variation across this range. And one thing you might notice, just to start, is that in general, bigger circles are a little redder than the smaller circles that they're next to. And that you might wonder, looking at that, if there's maybe a little bit of a effect of the level of human population density on Aedes aegypti's preference for humans, which would make sense. And sure enough, we did find that mosquitoes from urban centers were more willing to bite humans. When we remove sort of the broader scale regional effects, we find that that human population density explains about 20% of behavioral variation in our data. We can vary the radius that we use to calculate human population density to sort of see what scale they're adapting on. So it's like, are you looking at human population density in terms of microhabitat? Like, are they living right around people in a household, or are they living around people on a more regional scale? And so you can change how you draw that circle to calculate human population density from existing geospatial data sets of human population density. And we get a clear best fit for a radius of 20 kilometers. So that's where we minimize AIC, which gives us our best fitting model. But although urbanization was a major explaining factor, we actually found that climate was more important than urbanization for explaining behavioral variation in our data set. So that was 18% of data. But when we tried to explain those more regional effects, those more spatial effects, we found that there were two main bioclimate variables that best explained preference for humans. One was precipitation seasonality. That's the coefficient of variation of monthly precipitation. And then the other one was precipitation in the warmest quarter. And we can put those together into a sort of single index of the intensity of the local dry season. So I was telling you, some places are really seasonal, some places are sort of aseasonal. If we put together these two bioclimate variables into an index of dry season intensity, we find that explains 65% of behavioral variation in our data. And so in particular, I've shaded here in the hatched region corresponds to the highly seasonal Sahel region, just south of the Sahara Desert. This is the most seasonal part of sub-Saharan Africa. And in that hatched region was where we found sort of the reddest tones, the strongest preference for humans. North of that in the Sahara Desert, it's not very seasonal, it's green, but that's because it's consistently, there's no precipitation, there's no Aedes aegypti there. South of that, that sort of green area is the Congo rainforest ecosystem, where it's very aseasonal. So Kakamega, Kenya is on the eastern edge of that. Yeah, and so in particular, we found strong human preference in Aedes aegypti populations in places like this, in Ngoye, Senegal, where there's not a lot of natural habitat, but there's this great habitat living with human hosts in large human water storage vessels. Now, this isn't like for drinking water, it's more like a gray water for cooking and cleaning, and so it's allowed to be a little bit dirtier than drinking water would be, and it's really hard to fully empty and clean these out because they're big, heavy vessels. We found a lot of human specialist Aedes aegypti living in vessels like these. Now you might say, okay, you only have a couple of populations sampled from that Sahel region. Are you sure it's not just some other, some other confounding variable that's making you think that Aedes aegypti and these seasonal habitats are more specialized than humans? So to look into that a little more carefully, we collected a whole bunch more colonies from across that Sahel region. And sure enough, we do find lots of preference for humans from across that highly seasonal Sahel habitat. So our models seem to be working pretty well. Now, human population density and climate are changing rapidly in Africa. So how should we expect Aedes aegypti to respond? Well, we can take those two main structuring ecological factors, dry season intensity and human population density, and plot them out in this sort of two-dimensional parameter space where we color the background according to the projected preference for humans, according to our ecological model. And each point on this plot represents a present-day population with the behavior that we measured in our experiments. And so you can see that there's sort of a range of different values of these two ecological factors, and we can use publicly available UN projections of climate change and changes in human population density to try to understand where these populations are going in the near future. So if we look at what those projections say for 2050, we see that in terms of climate, the effects are maybe a little bit equivocal. So some places seem to get a little less seasonal, a lot of places stay about the same. It's not a super major effect either way. But if we look at the human population density effects, it's a lot less subtle. In the large and rapidly growing cities of sub-Saharan Africa, we expect to see a shift towards greater preference for human hosts in the next couple of decades. And that's because some of the fastest growing cities in the world are in sub-Saharan Africa. So that would take us from a situation like this, where we have a few pockets of human preferring Aedes aegypti in sub-Saharan Africa, and a lot of populations are generalists, to a situation like this, where in the really big dense cities, we see the emergence of greater preference for human hosts. Of course, we don't actually know what's going to happen when these populations move out of their present day combination of environments. Some of these projected population densities are much higher than what we've seen in the past. And so it'll be a really interesting thing to see in the coming decades what actually does happen. This is just more of a sort of an indication that we should be on the lookout. So overall, I've shown you sort of an ecological model that suggests that Aedes aegypti becomes more specialized on humans in places that have really intense seasonal climates and long, hot, dry seasons, and also in sort of rapidly growing cities. We think these climate effects were likely the original driver because those cities only reached their present day densities in the last couple of decades. And so it wouldn't make sense for those urbanization effects to have been the original driver of specialization, but they seem to be a recent phenomenon of growing importance. But to try to get a little more carefully at some of those questions, we wanted to use those genomes to actually test some of those hypotheses. So what are the genomic and historical origins of this behavioral shift? And to ask this question, I'm going to look at the 345 unrelated genomes sequenced to 15x coverage in collaboration with the 1200 Aedes aegypti Genomes Project. So first of all, if we just look at overall population structure in Aedes aegypti, this is what we get for our best fit model. So we use a cross-validation approach to ask how many sort of overall population clusters there are in Aedes aegypti. And we get a best fit value for three main ancestry components, and they are pretty interpretable. So there's this coastal East African component east of the Rift Valley. There's this west and central component in light blue. And then there's this red sort of human specialist ancestry component that's totally dominant in invasive populations outside of Africa, but present at different levels in different populations within sub-Saharan Africa. And sure enough, the more ancestry a population has from that red cluster, the stronger its preference is for human hosts. Now, if we turn up the sort of resolution of the structure analysis, moving away from that best fit value of k equals 3 to k equals 6 to try to subdivide populations further, we find that there's this sort of orange population, this orange ancestry component that seems to correspond to the most human-preferring populations in sub-Saharan Africa. And we think that orange cluster might have something to do with the origin of human specialists that then became sort of bottlenecked and gave rise to these red invasive populations outside of Africa. So we think that the origin of specialists might be in this Sahelian habitat, these highly seasonal habitats on the southern edge of the Sahara Desert, where mosquitoes do really well in human habitats, but there's not a lot of natural habitat. But there's just one problem for this sort of explanation, which is that Sahelian habitat hasn't always existed. And the best way I can show you that is with this petroglyph. So it's a carving that shows, I'm going to ask you to do a little bit of user imagination a little bit here, but shows people hunting big game on a rolling grassland in what's the present day Sahara Desert. And so it's a very different climate than a desert where you'd be hunting big game. And that's because it wasn't always a desert there. So from about 5,000 years ago to 15,000 years ago, there was this period known as the African humid period, where there was a sort of notable pause in the desert character of the Sahara, and we had instead a sort of a green Sahara period. And the best way I can show you that is with this record of Sahara dust. So every year, dust blows off the Sahara Desert, deposits in the Atlantic Ocean, and you see this sort of pause in the accumulation of Sahara Desert in the Atlantic Ocean from 5,000 to 15,000 years ago. So that's the African humid period. And so that sort of modern day Sahelian niche, where it's this really seasonal habitat north of a really inhospitable Sahara Desert, that's only existed for about the last 5,000 years. So we might suppose that human specialist populations of Aedes aegypti originated at the end of the African humid period in highly seasonal habitats. And we think that these human specialist populations spread to the New World along with the Atlantic slave trade about 500 years ago, 150 to 500 years ago. And then in the present day, rapid urbanization is associated, seems to be associated with increased preference for human hosts in that native range in sub-Saharan Africa. But do the genomes actually support this ecological model for Aedes aegypti's history? To answer that question, we can use cross-coalescent analysis. So this is based on basically looking at when different individuals shared common ancestors going backward in time, using the steady accumulation of mutations between lineages to date when they split. And in particular, I'm going to tell you about a cross-coalescent analysis of divergence between generalists and human specialists in Senegal. So there's a human specialist population in Ngoye and this generalist population in PK10. If we do a cross-coalescent analysis, looking at how these populations have split, it looks something like this. We have this signature of what's called cumulative migration, which should sort of steadily rise and plateau up to 1 when populations have merged into a single ancestral population going backward in time. And we do have this nice kind of like gradual merging process going backwards. But we don't actually know off the bat how to convert that into historical time. So there's this split, but when was that split? We know it's related to generation time and the mutation rate, but calculating the exact coalescent scaling factor is a famously difficult problem, even in systems like humans. But we can calibrate it using a much better understood sort of event in Aedes aegypti's history, which is its spread to the New World during the Atlantic slave trade. And so I'm going to tell you about another cross-coalescent analysis looking at the relationship between populations in Santarém, Brazil, and human specialist populations in Ngoye, Senegal. If we look at those two, if we do a cross-coalescent analysis of those two populations, we see a strong pulse of migration from in between Senegal and Brazil. And we know that that pulse almost certainly corresponds to the transatlantic slave trade due to the historical and epidemiological record. So if we put those together, we can get an estimate of a coalescent scaling factor that tells us how these coalescent analyses relate to historical time. But the question is that coalescent factor that we get, is that scaling factor biologically plausible? Well, we know it depends on the mutation rate and the generation time. Now, we think the mutation rate should probably be, something reasonably similar to other insects. So Drosophila melanogaster is around 3 × 10⁻⁹ per base pair per generation. Homo sapiens is a bit higher. We might think that Aedes aegypti is sort of something similar to, or maybe a little more than Drosophila melanogaster, based on published values for insects. Now, we think the generation time is probably somewhere between 10 and 20 generations a year. So we have this sort of like box that corresponds to the plausible values of that coalescent scaling factor. And sure enough, if we use the Bhattacharya coefficient to quantify the level of correspondence between the historical record of the Atlantic slave trade and the migration pulse that we saw in our coalescent data, we find that there's a really strong correspondence between these two signals. And so we can carry forward the best fit coalescent scaling factor and use that to understand the older relationship between generalists and specialists in Aedes aegypti's native range. And so if we take that scaling factor and apply it to our analysis of generalists and specialists in Senegal, we find that divergence really does seem to really, like, the generalists and the specialists split sort of dramatically right after the end of the African humid period, with the drying of the Sahara Desert and the origin of that modern niche in the highly seasonal Sahel with settled human societies living there. So we see strong evidence that the generalists and specialists split about 5,000 years ago with that sort of drying process. So I told you about the origin of human specialists and how they spread to the New World, but what about modern urbanization? What about that effect we saw where in big cities, Aedes aegypti was becoming more specialized on humans? Well, we can't really use coalescent analysis for that because it's too recent of a process, and so it takes time for mutations to accumulate, and it's just not really clear. But we can use a different approach based on the action of recombination over time. And so if you imagine that there was a pulse of human specialist ancestry, basically some human specialists came into a generalist habitat. In the first generation, hybrids would carry one chromosome from a human specialist and another chromosome from a generalist parent. After a generation of recombination, you might have these long chunks of chromosome coming from a specialist or a generalist background. And as generations progress, you get smaller and smaller chunks as recombination breaks down these tracts into shorter and shorter lengths. And so we can use the characteristic length of those ancestry tracts to estimate how long ago specialist ancestry was introduced into these rapidly growing cities. And we do find these sort of characteristic lengths of human specialist ancestry in both Kumasi and Ouagadougou, two of the most rapidly growing cities in sub-Saharan Africa. If we look at the human population growth in these places, we see that it really took off in the last 20 to 30 years. And the length of those ancestry tracts, shown in pink on the left, correspond to an influx of human specialist ancestry about 20 to 30 years ago as well. And so it really does seem like there was an influx of human specialist ancestry much more recently into these rapidly growing cities when they reached their modern sort of metropolitan character. So overall, we see a remarkable concordance between historical, ecological, and genomic evidence, suggesting that the human specialist form of Aedes aegypti originated at the end of the African humid period and the origin of the modern Sahel. It spread to the New World along with the Atlantic slave trade, and that in the modern day, rapid urbanization is associated with the rapid evolution of increased preference for humans. So I hope I've convinced you that intense dry seasons and dense human populations are associated with specialization on humans, that these human specialists likely first arose with the formation of the Sahel, that the global invasion seems to have been kicked off at the beginning of the Atlantic slave trade, leading to the emergence of yellow fever, dengue, chikungunya, and much more recently Zika. There's rapid contemporary evolution of increased vector capacity in urban sub-Saharan Africa. So basically they're becoming more specialized on humans. And overall, yeah, thank you very much to all of my collaborators who worked with me on this. I'd like to thank my funding sources, and I'd be happy to take any questions.
All right, thank you so much, Noah. I just want to remind everyone listening, if they have questions, please put them in the Q&A and we can get to them now or at the end. Or if you think of something late, there's also time to do questions on the Slack channel after the symposium is over. So right now we have two questions related to your methods. Brian Rector would like to know how many different hosts you tested versus humans in the tube assay, and did you test any non-human hosts versus each other?
Yeah, that's a great question. So most of the experiments, for logistical reasons, were done between one specific host who was a human host, which is me, and one of two different guinea pigs. But we get the same effects, the same rank ordering of preference for humans versus animal hosts, no matter what human host or animal host we use. So we can use, for example, a rat or a quail, like some very different non-human hosts as the non-human, and we get the same responses. Similarly, if you use a different human, like lots of different people, we get the same responses. Now, you can if you put two different people, like usually one person is more attractive than the other. I haven't done animal to animal comparisons. That's a really interesting question. I haven't looked at that, but I wouldn't be surprised if there are differences in the relative attractiveness of different animals, even if it's just about how smelly some animals are relative to others. But the main sort of like human specialist to generalist continuum is sort of recapitulated no matter which human we use, no matter what animal you use. It seems to have more to do with distinctive odorants that are sort of characteristic of humans, that make humans a bit weird as opposed to other animals. We have certain odorants that are more common in a human odor profile, like there's sulcatones, certain aldehydes, there's things like that that are a little bit characteristic of how humans smell, and the human specialists are particularly like cued into those.
Okay, great, thank you. And I see that Anna Childers would like to ask a question live. If you want to go ahead and ask, Anna.
Oh, no, there was a chat question that was put into the chat. They said, Noah, such a beautiful talk. Regarding the ancestry track analyses, have you looked at any associations with specific regions or genes for human preference? And that was a question from Amanda.
Yeah, that's a great question. So I'm really interested in going more in that direction, because you're totally right that these like short tracts of ancestry like if you can get enough individuals and they have different parts of their genome that come from the human specialist or the generalist background, you could actually do some fairly high resolution trait mapping analyses to try to associate specific genes with, for example, preference for humans. I haven't gotten to that point yet, basically just because it's a logistically complicated experiment where we need to get a whole bunch of mosquitoes into an olfactometer with the right kind of admixture profile to start to look at specific genes. I mean, if we just look at like where you have more human specialist ancestry or less in the genome, there are certain parts of the genome where we see it sort of like more concentrated in a way that suggests that it might be under selection. But those regions are fairly large, so it's hard to associate those with specific genes, like this gene is causing a behavioral difference. So I think we'd have to do a more careful sort of follow-up admixture mapping experiment. But I would be really excited to do that, yeah.
Very cool. We have a new question that's been, oh, Amanda says she's looking forward to the follow-up on that, by the way. We have a new question now that's asking if this recent human colonization is the result of contact between human specialized populations and or convergent evolution specialization.
Oh yeah, so I if I had more time to talk, I could have talked more about the sort of more broad scale genomics, but the answer to that question is that it seems to make use of the same genes. So there's a single underlying ancestry component that's associated with preference for humans wherever we find it. And it seems like the same sort of genetic signature is associated with that shift every time. So it's not like they're coming up with new solutions from scratch. It's like they make use of the same alleles, the same genetic variants to make the shift when it happens, either historically or in populations that are making that shift in the present day.
All right, and then Brian again has a question about your testing methods. Have you tested non-human hosts that are native to Africa?
Yeah, so let's see, like we've looked at, I mean, so I'm trying to think, I've looked at guinea pigs, which are not, rats and quail, which I guess, I mean, rat, that's the Norwegian rat, right? So I guess that's not native. Quail, that's a domestic quail, so probably not native. I've looked at sheep's wool, also not native, although there's like some native, definitely some native like goats and sheep that probably smell similar. So I haven't done like a super controlled like what is the most natural host for the generalist form, and that would be really interesting to do. I would be curious to look more at that. The fact that we get similar responses for very like taxonomically divergent non-human hosts suggests to me that at least I would be surprised if there were big differences between, like, for example, a native African rodent and a rodent from another continent. If we see the same response for like birds and rodents and also like ungulates or something, I would be surprised if it matters whether you have the native or the non-native one. But that's a totally good question. I think it would be good to look at a broader variety of animals. There's actually a graduate student in the McBride lab, Jessica Zung, who has a really cool thesis looking directly at some of these questions, like what is the taxonomic distribution of like different odorants, and what does odor space look like compared to taxonomic structure? How is like how animals smell related to their phylogenetic background? All those kinds of questions. It does seem like there's kind of a distinctive human smell. Human smell a bit weird compared to other animals for a variety of reasons. And so I would be really surprised if there was a different response, but it's a great question. Like I haven't explicitly looked at responses to like animals that were chosen to be representative of the most ancestral kind of host for these mosquitoes. Yeah.
Okay, great, very good. Well, if anyone comes up with more questions they'd like to ask, feel free to continue to add them to the Q&A, and we can always check back with Noah at the end of the session. So thank you, Noah. That was a wonderful talk. And we will move on to speaker number 2. The next speaker is Zach Cohen. He's a postdoc actually in my lab at ARS here in College Station, Texas. And he's relatively new, so he's going to be talking today about some brand new data, which is always exciting to learn about. So with that, I'll let Amanda cue up the talk.
Good morning. Thank you for that warm introduction, which I presume occurred My name is Cohen, and I'm going to be presenting some very preliminary data on the system, the boll weevil, which we hope will be well utilized by the field in general and offer some interesting insight into this notorious animal. So my talk is entitled Whole Genome Synteny, Weevils, Gene-Rich Inversion Between Anthonomus Pest and Non-Pest. Apologies for that. That is a picture, a cartoon of Elmer Fudd, who famously wolls his R's. Alrighty, so the basic boll weevil, Anthonomus grandis. Well, you'll notice right away that it is a trinomial nomenclature, which is a degree beyond the classical rank of species. And that's because there is another subspecies, Anthonomus grandis thurberia. And the difference between these insects is that grandis has co-evolved and prefers commercial cotton, the cultivated cotton variety, as opposed to thurberia, which has a preference for the Arizona cotton or the desert cotton, Gascipium thurberi. This map shows the expansion history of the boll weevil from the southern tip of Texas at the end of the 19th century, all the way to southern Virginia, the Carolinas, in the 1920s. And the colorful plates on the right show the life history of this bug. It's missing an essential stage, the eggs. Obviously, these insects do lay eggs either on the square of the flower or after pollination, the boll that gives rise to the cotton ball, the fruit. And this depends on the number of generations in the field and the timing of the volatinism and the flowering. Now, this is very similar to another insect near and dear to my heart, the Colorado potato beetle, which of course is native to North America and kind of also expanded eastward using the potato plant. These two insects are very, very similar. So these are the host plants of the subspecies grandis and thurberia, respectively. In case you were curious what a cotton plant looked like in the field, this is sort of right before harvesting of cotton balls. And yeah, it looks like someone's Etsy project, but that's it in the field. And obviously this is very phenotypically different than the ancestral wild cotton, which is a tree. This is Gascipium thurberi here, and you'll see on the left here in this lush sort of valley, and these are the flowers, not the cotton balls of the plant. And again, that's the host plant for Anthonomus grandis thurberia. So I say that this insect is simultaneously reviled and revered. Obviously, it has this reputation given its impact on cotton production in the southern United States. Again, in the beginning, the first half of the 20th century. Now, the silver lining is that it is revered in some communities, some agricultural areas, because it offered a novel niche space for alternative crops to be grown. George Washington Carver was really essential in transitioning from cotton production to another very delicious and popular crop, a legume. Anyone? Yes, a peanut. And that's what this proud farmer has in his hand, a peanut plant. That's also what this statue on the left here represents, the sort of celebration of this boll weevil, of ridding these farmers of cotton, such a pain, and sort of providing the motivation to transition to peanut which is in Enterprise, Alabama. One day, if I'm fortunate, I will make a pilgrimage there. So what is the utility and some of the motivations to have a whole genome sequence for this insect? So when it comes to the sort of applications of this resource, we want to be able to distinguish the boll weevil from other weevil species that are morphologically similar and hard to distinguish, with the naked eye. Want to understand kind of where new boll weevils that are captured in a field might be coming from. The U.S. eradication effort has been very successful, which I'll discuss a bit further. But there still are reinvasions in eradicated zones, so determining the origin of those beetles is important. And of course, this insect has been a target for control using various insecticides over the years. It had developed resistance to DDT and some pyrethroid formulations, but is actually fairly susceptible to malathion, and which is an organophosphate, which has been used in the field for the last 40 or so years and is still 100% effective against the insect. So kind of unraveling or determining the sort of genetic associations that could be describing this maintained susceptibility is interesting. And I had mentioned that these insects are caught in the field where they're lured using a pheromone blend, a bait. So understanding the gene sort of structures of chemosensory receptors, of odorant receptors, could help develop a more precise blend to capture the boll weevil, distinctly from other Anthonomus that might be in the area. And of course, some of these more basic, intriguing things to pursue, these motivations, understanding just the larger genomic context of this insect, larger polymorphisms, structural variations, inversions, etc., and reconstructing the phylogeny, having a reliable method to sort of determine, derived clades within this genus. So I've mentioned this eradication history. Here's a timeline of the expansion from southern Texas throughout the South, and the sort of response by the state and growers. So by the early 20th century, the whole field had sort of been developed around understanding the boll weevil and controlling it, which led to the development of these pheromone baited cones and the implementation of this statewide eradication effort. So there's a zero tolerance approach when it comes to controlling this insect. If there are any boll weevils at all in the field, it'll sort of necessitate a major control effort. So fortunately, or unfortunate for this type of insect, this native species, 98% complete. There are a couple zones still in southern Texas in the Rio Grande River Valley where control of the boll weevil is just part of the cotton agricultural production. And Texas is the largest cotton producer in the US. This is a map kind of showing the statewide eradication effort. The blue and green emphasize zones that eradication is ongoing or has been complete. And you'll see there's also some northern Mexican states represented here that kind of still have chronic infections, or they're not a part of this program. This map is old. I don't have a more recent version of it, but these colors have changed somewhat. So what are some of the roadblocks to weevil eradication. Well, as we've said, this insect is native to the area. It's co-evolved with these plants and in this environment. Of course, the geopolitical border between United States and Mexico is sort of irrelevant to this insect. So determining where reinfestations occur is important in understanding if they're migrating up from Mexico or if they're native populations in the southern United States and southern Texas, for example. And that was relevant for two recent reinfestations that occurred in previously eradicated zones of Texas. So Uvalde, is fairly north of Mexico and had a major outbreak, a reinfestation. 2015, it was a 4-year event, and it was pretty costly for the state at the time. Kingsville was more recent, not as costly, and both of these reinfestations seem to be from individuals derived from Texas, so from the Rio Grande River Valley. And there were genotyping efforts that determined that. So what are the strategies to collect and control this insect in the field? So again, these are the two subspecies that are often confused with each other. And the first step is the implementation of this pheromone bait, this lure. That's what you see here in chartreuse green. You'll notice that this chromatogram has 4 distinct peaks. I couldn't tell you much about the chemistry of these active ingredients in this pheromone blend, but they're appealing to Anthonomus generally. So you're getting multiple species kind of being captured in this lure. Although, it is designed for grandis. That is what we presume is on this lure here, but without having, reliable, high discriminatory diagnostics, we can't make that assessment. So that's what the whole genome sequence data will hopefully provide. Is it a bull? Classical morphometrics were used to distinguish the two subspecies, grandis and Thurberia, which is fairly robust, 89%. Of course, there are morphological variations within these two subspecies, of course. And the CO1 profile, as we know, is a reliable marker of the mitochondria. It's a fairly large, region as far as bases are concerned. But this depends on time and resource allocation, which might be unfeasible in the field, a sort of like rapid diagnostic. And that sort of motivated the study by Razik et al. To generate a SNP with much higher resolution to distinguish these two subspecies. This particular SNP, 317, has about 98%. So ideally, with whole genome sequence data, we can, increase that number maybe to 100%, or use multiple SNP markers. So what is a boll weevil as far as the evolutionary history, sort of prior to cotton cultivation, pardon me? Well, there are two competing hypotheses. One that suggests the populations are geographically distinct from each other, given this Sierra Madre Occidental mountain range that sort of bisects their native range. And the third, the other hypothesis is a 3-source hypothesis, and this is associated with sort of land races and the host plants that they feed on. Again, you have the eastern and western subpopulations, but you also have a southern Mexican population that is indeterminate of either the Thurberia or boll weevil. So Razik et al. Had incorporated 200 or so individuals to sort of address this question using a draft genome assembly from 5 populations. Now, there wasn't a southern Mexican population that was used here, but these are representing Thurberia weevil and the cotton boll weevil grandis commercial weevil that are sort of sympatric within these two zones, eastern and west. So what they determine is actually it's not as associated with the host plant as it is with the geography. So there's a difference in the eastern and western clades. Now, again, this is an unrooted phylogeny. Having a reliable outgroup will help sort of root this tree and determine the derived clades, which, you know, is relevant and a little bit more reliable in distinguishing sort of the sequence of events here. So once an insect has been determined to be boll weevil, you know, or not, in the Rio Grande River Valley, they are just constantly applying malathion applications. And you can see this is throughout a growing season a couple years ago, pre-demic. And you'll notice that, or really right in the beginning, tens of thousands to, in August, 120,000 applications of malathion. So quite a huge load of this chemical in the field to control the weevil. So kind of understanding a bit more about the genetics might contribute in reducing that burden for the farmers and the bug. So what are the efforts to date? So now we're going to kind of transition into the main feature of this talk. I was referring to the 2021 Razik et al. Paper. They had generated a draft assembly that was fairly decent. Since then, our group with the Ag 100 Consortium, a big shout out to Dr. Shenna Sem, Anna Childers, and Scott Guide for their effort on this and helping us you know, push this assembly through and meeting all of our criteria. Dr. Amanda Stocky has also been helpful on that, so thank you. So one of the major discrepancies between the genome assembly size we were producing and the C value that we were generating based on the picogram weight of the nuclear DNA was about 200 megabases for grandis. And actually the Thurberia assembly was a little larger than the molecular weight. Now, there might be some interesting features within these genomes that we're not capturing in the assembly. We thought perhaps there were significant repeat elements, or maybe falsely concatenated haplotigs based on heterozygosity. This is still outstanding, more or less, but we do have assemblies that are good enough. You know, every other metric is looking good, that we're moving forward with, and that's kind of what we're doing. But we haven't really resolved what this difference in size is. Furthermore, these two samples that we have kind of represent the eastern and western lineages, which is useful for our work here. So one of the first things I did was sort of quantify the repeat element, you know, share distribution among these two or between these two assemblies. And you'll see that the distributions are fairly similar, especially, you know, in the total interspersed repeats, transposons, etc. And this was carried out using the repeat modeler, repeat masker pipeline. You know, it's just like pretty straightforward. And from here, I used the grandis draft assembly for annotation with 48 or so individuals that we had RNA-seq data from, as well as the protein Arthropoda OrthoDB set. So multiple lines of evidence were incorporated in Breaker, which is what it's for, which is nice. And from there, we used these gene model predictions into BLAST. And this number, 17,500, represent the blasted and interpro annotated gene models, which is right on par with most other high quality assemblies, especially within Coleoptera. And this fell on 80 scaffolds, which is nice, of 140 or so. Of these, though, about 3/4 had a unique start site. So I curated by start position per scaffold, and approximately 25% of these annotated genes are probably paralogs, which is relevant for copy number variation, evolution, and potentially some other downstream investigations. The next step that we wanted to work on was to identify large-scale synteny between these two assemblies. And you'll see generally that they are highly syntenic with one another. The largest scaffolds with the largest. Some of these smaller ones are not mapping to large, which is nice, that they're probably some noisy artifact or repeat-rich regions. But you will notice that there is this large inversion right in the middle of the plot here. So I wanted to understand a bit more about this inversion, determine the start and stop sequence, as well as any annotations that resided within it. I use Minimap here to query these inversions based on the coordinates of the reference to the query. So the reference is a grandis, query was Thurberia. And you'll see that the coordinates of these inversions on the right here kind of coincide with the dot plot, where it's like the beginning of the grandis scaffold, and then maybe these two terminal residual ends here are inverted sequence. But actually, the positions of these inversions with respect to Thurberia were largely continuous. So that was kind of unusual. We're not really sure what to make of it. If anyone has seen this before or can offer some advice as to what might be explaining this, maybe I did something wrong. It's very possible. The parsimonious explanation might be, well, there's one inversion on Thurberia as opposed to two on grandis, so perhaps that's the source of the inversion. We wanted to identify the source without using an outgroup because we don't have one right now, and without having a sequence like the breakpoints, which of course is a tried and true method. But if anyone, feel free. So using the coordinates of the inversion and the GFF, we determined genes that were within these large inverted sequences. It was about, I think, 17 megabases, so fairly large. And there were 250 annotations. We did a gene ontology enrichment. That's what you'll see in this very useful, very relevant word cloud. Most of these are metabolic processes, at least at this perspective. We wanted to focus in further on some of the highly associated genes with detoxification, with chemosensation, transcription, and TEs, transposable elements. And you'll see that there were quite a lot of transcription factors, 23 in me. So it's curious to understand how they're influencing expression within this region. I think the presumption of such a large inversion is that obviously it's going to affect recombination, but it might also affect expression. The CYP locus was actually a mitochondrial locus, CYP-C in the C family, and this was only a partial hit. Now, the odorant receptor was interesting. This was a complete, start codon to stop sequence of OR29, and it annotated to two distinct regions within this arm. One on the duplication. So perhaps an inverted duplicate. Unsure what the biological significance of this is, but that was kind of interesting, maybe. And I wanted to try to identify or determine if these inversions were descriptive of the East-West two population hypothesis. So we used three populations that had more or less the similar number of samples, 44 and 41. So the Eastern was from Southern Texas and Northern Mexico. The Western Arizona population, which represent Thurberia, are from Arizona. This was incorporating this find inversions R script from Shenlin Liu, and you'll see that, again, it was a large region. The curation process of like 99% individuals in ANSD, reducing minor allele frequencies, et cetera, for quality control, resulted in only 1,700 loci. And there really wasn't anything obvious here. In fact, these were all insignificantly different from one another. This is the proportion of the inversion based on the SNP frequency. So ideally, SNPs within the same haplotype of an inversion are going to have similar allele frequencies. So this was like a sliding window approach. And I think given the low density of the SNPs and maybe the high curation, this is not imputed. There are a lot of variables that could influence this result, but this was just kind of a first pass. So perhaps there's something here, but right now it's insignificant. All right, so what are the future directions here now that we have all of these data to kind of play with? So of course, one of the first things I'm going to pursue further is this allele frequency inversion between the East and West populations. Identify discriminatory SNPs, as I said, maybe improving that 98% discrimination. The chemosensory and metabolic genes for baiting and control, characterizing gene expression. We have a lot of RNA-seq data of the boll weevil throughout its development and also throughout a malathion, time series dosing project. So characterizing gene expression throughout those processes is going to be fun. And obviously the litany of deep and recent time evolution, phylogenetics, which really gets my motor going, is going to be relevant as well using these resources. So I just want to thank everyone for paying attention to me at this point. Cotton, the fabric of our lives, ARS and USDA for this opportunity. Of course, the charismatic boll weevil. And my group here, this was taken prior to my arrival. I'm not in that photo. I wasn't taking it either. But these are the faces that I see and I associate with Research Fam. So with that, I will leave it to Real Zach, IRL, and thank you.
All right, thank you, Zach, for that great presentation. Some exciting new data coming in. Questions are kind of slow right now, so if anyone has questions, please add those to the Q&A. And if not now, at the end of the session. A few may be coming in via text right now, but I guess instead of waiting right now, we should move on, since we're short on time anyway, to our next talk, and then we can always circle back with questions for Zach at the end. So with that, I guess we'll move on to our third speaker, Perot from ARS. He's a bioinformaticist in Kerrville, Texas. He is giving the talk, but his partner in crime for this, for the data he's going to present, is Susan No. She is a USDA Animal Disease Research Unit stationed at Washington State University. So with that, I'll have Amanda queue up the next talk.
How does that look? Do you see the title slide?
Looks great.
Great.
Well, hello, everyone. Thank you for attending my talk. I want to thank AGSx for giving me the opportunity to give a presentation on some of the research that I guess I'm Perot, Saelao, and Dr. No have been working on. So the title of my talk is TickTok, a short video update on the comparative analysis of tick genomes sequenced within the USDA. Originally, I had just wanted to come up with a clever meme title and give it out there, but it looks like they called my bluff, and so now I'm giving a presentation. So everything that follows this slide may be nonsense, but it has been somewhat thought through now. So this work has been primarily led under the direction by Dr. Susan No. She's a ARS animal disease researcher located at Pullman, Washington. And her goal is to develop methods to prevent tick-borne diseases with a focus on Amblyomma anaplasma marginale, a tick-borne pathogen of cattle, which is found worldwide and has the greatest impact in tropical and subtropical regions. So her research efforts are focused at both the vector and pathogen interface, and thus that brings us to tick genomes because they play a big role at directing this pathogen and mammal host interaction. And a little bit about myself. I'm from the tick and biting fly unit, so I work at the Nippling Bushland U.S. Livestock Insect Research Laboratory here in Kerrville, Texas. And recently, our unit has gone through some rearrangement, and so right now, I'm actually a part of the Veterinary Pest Genomics Center, where our goal has been mostly to utilize biological resources and next-generation sequencing technology to help improve, annotate, and just generally drive better genetic and genomic research tools for those people studying livestock pests or veterinary or pests that are of veterinary importance. So originally this conception had started off as a center, but now we're a unit. So I think might be the only person in my unit, so this is the loneliest unit in the world. But looks like it's going all right. So, Dermacentor andersoni, otherwise known as the Rocky Mountain wood tick, is a 3-host tick that will spend 3 separate life stages across 3 different hosts. So for one life stage, the larvae to nymphs, they'll spend on one host. Then when they mature, they transfer to a new host and go from nymph to adults, and then subsequently lay their eggs on the following 3rd host. This entire life cycle can occur in just 10 weeks, but typically you'll see this across 2 to 3 years. Much of this is dependent upon the environmental temperature and moisture factors. So Andersoni will overwinter and emerge in the spring, and similar to what was shown in that previous picture, they'll climb onto blades of grass and attempt to latch onto their hosts. They'll target larger hosts, which include kind of deer, elk, horses, cattle, and occasionally people. And this usually begins, as I mentioned, in the early spring as temperatures increase. And conditions that are supportive of tick development can advance the timing of the life cycle and increase tick abundance. With continued climate change, however, earlier warmer springs may lead to adult tick questing earlier in the year, but may be countered by hotter summers. So regional variation in precipitation and temperature can have an effect of how often and where you see these ticks. So Dermacentor andersoni can transmit viral and bacterial pathogens to hosts, and importantly, they can transmit these diseases to humans and animals such as cattle. These include Rickettsia. I'm not a disease person, so I'm going to butcher those. But they also transfer anaplasma, which is the cause of bovine anaplasmosis. And this transmission of these intracellular pathogens requires direct interaction between the pathogen and the tick cells, meaning that the ticks have to bite the individuals to transmit these diseases. So what we have here is you can see that cases of anaplasmosis in the U.S. have been drastically increasing from the early 2000s up to present. And that even across age groups, just seeing higher incidences. I don't know what it is that being like 80 and wandering around in the woods means you get anaplasmosis, but it's probably more like you get bit and you get the disease because you're old. Anyways, I'm not saying that the disease is old. I hope that this doesn't make it into the final cut of the recording. So what genetic variation exists in the species history? Now, we there's been a huge lack of information on the genetic variability and population variation of this tick, solely because there was not a genome. So that was one of the main drivers of why we wanted to develop this genetic resource tool. However, there have been some studies that attempt to map phenotypic variation and genetic variation. So what you have here is a study in the early 2000s, which attempted to characterize the genetic variation of big, the B, and small ticks, and how that might actively interact with efficiency of disease transmission. However, we don't really know much, is the true answer. But what they were able to show is that, with these big and small individuals, and when they crossbred them, that there was a wide variety, that there was a big range of different phenotypic variations that exist, but was there any genetic variation that could explain this phenotypic variation? And actually, what they ended up finding was that when they did 16S and ITS2 sequencing, now this is somewhat older technology, they found 9 different haplotype blocks, or 9 different haplotypes related to these 2 body size phenotypes. However, when they looked at the 16S gene, they found nearly 14 different haplotypes detected, showing that there were a tremendous amount of genetic variation just between these 2 phenotypes. Now, in other studies in Dermacentor hunteri, it was estimated that there are only around 11 different unique haplotypes for the 16S DNA gene, and other species such as Scapularis only showed around 7. So it's really interesting that D. andersoni had so many different haplotypes found solely just based on this big versus small, whereas other tick species have shown far reduced genetic variability with respect to something like this phenotypic variation. So another study looked at another region of the 16S gene across 6 different populations collected in Montana, and what they were able to find is that there were no or little to no separation among these individual populations based on the 16S gene sequence. So that is a little bit in conflict with what was previously reported, where they found just, quite a bit of genetic variability in the 16S subgroup. But this study here was actually showing that there was no consensus and that they aren't able to place them into unique population subgroups. Well, I guess that's actually meaning that there is so much genetic variability that they can't classify them into distinct groups. So that kind of leaves that open-ended question, which is what we're hoping to eventually address with this new genome is, can we improve the genetic resources available to better understand the pathogen interaction, tick-pathogen interactions and disease transmission, and support our lack of knowledge and limitation of information in this idea of genetic and genomic population diversity. To do that, you always have to thank the people who help you. We partnered with the Ag 100 Pest Initiative. So I guess that name is a little outdated now because they've sequenced by far more than 175 species. So they've been a great resource in terms of sequencing and getting this genome off the ground. So I won't probably go into too much because I heard that someone else before me is going to be talking about this. But this species was selected as one of the top 100 ag arthropod pests that required support and needing the sequencing for this project. And the overall goal of Ag 100 Pests is to bring these genomes from millions of pieces or very fragmented arthropod genomes that have not too great of quality to high quality genomes which can actually be usable. So as someone who uses quite a bit of animal and insect genomes, it's great that there's an initiative that's going to pull these genomes out. And so the way they do this, and I think after talking to them quite a bit, it sounds like it goes from the wet lab, and then they do the genome assembly, and then it goes into the cloud, where they joke that, it goes through quite a few iterations before you finally get the filtering and submission. So, and I don't know, they have probably mentioned in the past that they are doing the submissions to to NCBI. And actually, this genome, probably by the time this talk is presented, will have been submitted to NCBI. So that's great. So on to the actual genome. Tick genomes are amazing. They're huge. So I'm sure that maybe I'm speaking to the choir, but this genome, it looks to be nearly 2.8 gigabases. And, shout out to Dr. Sim now over there in Hawaii. She did an amazing job of assembling, this genome and putting it together. And it looks like, the BOOSCO score alone shows that it has a 94% completeness. So we're getting an outstanding N50 length, showing that generally many of our scaffolds are complete, or at least largely complete. And this is at the contig level, it hasn't even been properly or completely scaffolded, if I'm to understand that correctly. So in addition to assembling the genome, we've managed, and I say we, once again, I point mostly to the Ag100 pest people. They've also helped us to assemble the mitochondrial genome. So we have now the information nearly complete of the entire mitochondrial genome. So this is going to be one of the amazing tools that's going to allow us to do a proper, mitotyping of individuals for better future population discovery and population expansion experiments when we have, more sampling and better research towards those kind of questions. So when we look at the dermacenter genome and compare it to the only other dermacenter genome published accordingly, which is Dermacenter sylvarium, we see that we actually get a pretty comparative quality to the genome. And similarly, the genomes of are of roughly the equal scale, 2.8 to 2.5 gigabases. So once again, these tick genomes are just enormous. But, the sylvarium genome is a great genome, and it's been brought up to the scaffold level. And we see that we get, nearly identical quality in comparative metrics. And hopefully we can begin to, as the genome gets used, curate it more and compare it and curate it further as we go along. So in addition to the Dermacenter andersoni genome, when we break it down to the contig reference, which is what the current quality of the genome is, we see that our metrics kind of come a little bit more closer in line. But overall, I think it's just to highlight more that we're seeing an outstanding quality of the genome, and that they did a great job. So in addition to the Dermacenter genome being published or being developed, Ag100 Pest has also taken on sequencing the Haemaphysalis longicornis genome. And so what I have here is the Andersoni genome compared across the published Haemaphysalis longicornis genome by a group, a Chinese group, and the one that's going on at Ag100, which is the research strain that we typically will use. And what, once again, this is to show is that the genome is of an outstanding quality. But what's interesting is that you see that the Ag100 pest genome has, nearly double the size. So I think we suspect that there might be some sort of triploid, polyploid genome thing going on there. So the longicornis has a pretty interesting behavior, or I guess physiology, where they have the potential for being beyond diploid. So that'll be really interesting to look into in the future when we start to get that finalized genome published or up on NCBI, and we can do a deeper dive into the analysis of the data there. But so now that we have a genome that's more or less an amazing genome, what are we going to do with it? So the whole goal of this group is not just to publish genomes, because that wouldn't be the only reason we want to do that thing. But we also want to look into what are the functional aspects of physiology that we can extract out and information that we can expand upon now that we have this resource. So hard ticks have and the next few slides are going to just be kind of a case example of what we could potentially do, or what we can do and have done, with this genome and what we can expand upon in the future. So hard ticks have a long feeding period. Blood digestion can be divided into 3 phases, and in addition to that, it's a long process. So the digestion process is also unique in ticks because it occurs intracellularly within digestive cells. So blood meals are typically taken up by the tick, and then it is taken up by digestive cells. And the blood meal digestion occurs in these cells through proteolytic enzymes. So most of which have target the hemoglobin molecule at a specific site to degrade and digest. And these breakdown of products actually build up in the tick, and ticks can't really excrete when they're up on or hosted onto animals. So it's interesting that they'll actually sequester many of these heme breakdown particles and in specialized vesicles. So anyways, just some fun physiology facts. But this process is actually run by quite a few genes. Specifically, there are these FER1, DMT1, IRP, fetch, which play a big role in regulating this process. Now, there has been a lot of study onto how, what do these genes do and how do they actually functionally impact the host. And so now that we have the genome, we can begin to look into this. So I'd like to take credit for this, but this isn't my data. But what we see here is that those genes, and so when you are iron deprived, those genes actually decrease in expression or begin to turn off. So we're showing that more and more that these genes, FER1, etc., play a big role in iron metabolism, and that, not surprisingly, these genes are important in metabolizing iron and iron-related cellular processes. So now that we have the genome, I was able to extract many of these genes, and starting with IRP1, and compare it to the various other Ixodes species. So what you have here is a neighbor joining tree to depict the sequence that I harvested from IRP1 compared to all the other Ixodes related species. And so unsurprisingly, you see that Dermacentor andersoni is IRP1 is really closely related to the Salvarium. But then from there, you get a pretty wide distribution of how the Dermacentor's IRP1 compares to that of the other species. Now, other studies have shown that many of the genes that are conserved between Dermacentors are very much conserved because of the processes that they play, and how you see different factors such as the 3-host tick behavior, in addition to Dermacentor andersoni's very broad feeding habits across multiple species as a factor that might be driving genetic variability and differentiation from the other ticks. So, and in addition to IRP1, we also looked at DMT1, which once again had far fewer isoforms. So you don't see as big of a branching tree within the phylogeny. And once again, you still see that Andersoni and Salvarium are much closely related. What is maybe more interesting to me, and I was a little bit more surprised by, is that that the Rhiphocephalus species and the Dermacentor species share more commonality compared to the Ixodes. Maybe that's not as surprising because I think Ixodes as another type of tick that they do perform different behaviors, but they're both blood meal feeding. So I guess I wouldn't have expected as much variability within blood meal feeding on a gene such as DMT1. Fetch 1, once again, has far less variability and are much more centered, and actually, interestingly, less annotation and sequence identity that's been known. And so what we see here is, again, that you don't have, unfortunately, in a lot of the Rhizophilus species, able to identify fetch sequences, but we're able to fish out the Dermacentor Andersoni gene and compare that to these species. So lastly, one of the most important genes, not this one, sorry, ZIP13, once again, and you see a lot of different isoforms of this gene. So this is more of just a depiction of just how much variation you can see in a single gene such as ZIP13 compared to that of the other species. But what I wanted to kind of get into here is FIR1. So FIR1, there are roughly around 52 available arthropod ferritin genes or annotated sequences that are grouped into two kind of distinct lineages, LCH type and HCH type. Now, that typically relates to the systolic functions of these type of ferrins. And so what we ended up seeing is that Dermacentor Andersoni, along with Variabilis and Albopictus, all clustered together, which is great because it's Andersoni. However, another previous sequencing of Dermacentor Andersoni didn't look like the one we found from our genome, but that might be because of genetic variability. But I get beyond the point. Variabilis actually has a 99% sequence identity to the Andersoni genome, so suggesting that the Dermacentor Variabilis FAIR1 sequence conservation is very tightly, correlated or similar to that of Andersoni. But all 10 of the tick ferrins, along with all these other HCA type genes, have been found in ticks. And so I guess essentially what this is describing is that this is a very highly conserved gene, but you're seeing a tremendous amount of sequence conservation between Andersoni and Variabilis. So there's something that's some process of blood meal feeding specific to FIR1 that's conserved between these two Dermacentor species. And so in addition to being able to extract functional sequence species gene sequences, we're also able to look at syntenic relationships. So once again, we only had Silvarium and Andersoni. And so forgive the labeling on the one, that's actually incorrect. And hopefully I'll have time one day to go back and correct that. But scaffold 30 of the D. Andersoni, when we look at syntenic relationships to that of D. Silvarium, so Silvarium has around 11 chromosomes. Interestingly, a lot of D. Andersoni's scaffold 30 shares similarities between those 11 other chromosomes. So this is the colors relate to chromosomes 1 through 11, and that top green bar here is scaffold 30. And so you see that there's a lot of sequence conservation across the scaffold 30 to that of chromosomes 1 through 11. So, and scaffold 11 or scaffold 30 is where we found the FIR1 gene. So perhaps there's something about that region, and once again, this is a scaffold and it's not complete yet, so it's not to the level of, like, say, a chromosome, but what we see is that there's a tight amount of sequence conservation on that scaffold according to the other 11 chromosomes that's shared. So anyways, that I think is something that could be potentially really interesting to look for into the future, is more of the syntenic relationships between these different species and how the relationships of these various genes are conserved and kept between them. So, well, thank you for listening to my rambling. This is a pretty pathetic thank you slide, but I wanted to thank again Ag 100 Pest or Ag Pest 100, specifically Shayna Sim, Scott Guy, Amanda Childers, Kevin Hackett, and the rest of the group. PacBio and their contribution to a lot of our work has not been underappreciated. And the rest of the scientists at the La Peroux Lab group. So, and again, AGSx and you all for attending my talk. So with that, I'll be happy to take any questions, and I appreciate your time.
All right, thank you, Perot. That was a great talk, and we do have some questions coming in. There was also a question for Zach at the very end. We'll circle back. I just wanted to put that out there so I don't forget. All right, so Perot, Meg Allen was curious if polyploidy is documented in other ticks.
I'm not quite sure if it's in other ticks, but I know that with H. longicornis, there's been known I think it's parthenogenics. So that kind of interesting chromosomal ploidy has been observed in there before. So it's interesting to see if that will, I guess a better insect biologist than myself would answer that and say probably, but I think it's been documented at least in H. longicornis for that interesting ploidy.
Yeah.
Okay, I see Susan's on. Do you have something to add?
Just to reiterate kind of what, add a little detail to that. There's, with Hemophysalis longicornis, there's two kind of big populations. One is parthenogenic, and that, at least in other populations, has been triploid. And then there's I don't know the word for it, but the population that has, males and females that need to breed to reproduce, and those tend to be diploid. That's the extent of my knowledge of that.
Okay, we have.
And to add, I guess, the population that is invasive here in this country that was introduced is parthenogenic. So we would expect this one to be triploid if that holds true of the other parthenogenic populations.
Very interesting. Okay, so we have another question. First of all, great talk. This is from Jeff Grabowski. He is asking that once your tick genomes are more finalized, example, assembled, annotated, are there any thoughts or plans to upload this data into Vectorbase for community use?
Yeah, that's a great question. And I muted the talk, so I couldn't hear myself. So I'm assuming it went well. But yeah, I think so. I think in the end, this is a community resource. Once we created the, I think Ag 100's goal as well is to kind of create this resource for the scientists to use. So I think Vectorbase is great. I've used Vectorbase before, and I found it really amazing. So anyways, I would love this resource, and I hope that I speak for the group members that I think it would be a great addition to Vectorbase. And yeah.
And I'll just read what Anna Childers put in the chat here. Yeah, this is going to be supported by I5K Workspace as well as Vectorbase. The Ag 100 Pest team is actually working on genome assemblies for a total of 10 tick species. And then she adds to the discussion about Hemophysalis longicornis. There are triploid, diploid, and aneuploid populations of H. longicornis. Triploid or parthenogenic, diploid requires sexual reproduction, and our sample is triploid.
Okay, good. I see someone had their hand up. So participants, I can't unmute you, unfortunately, to do live questions, but if you could put your questions in the Q&A, I can read those aloud for you. So I'll give that person a chance to write their question. In the meantime, I'm going to circle back to Zach. You had a question from Brenda Oppert. She says, nice talk. Can you be sure that the inversion is not an artifact of genome assembly? Is it on a large scaffold or contains multiple scaffolds? Did you have any long reads that bridge the ends of that inversion site?
Yeah, I saw that. This is a great question. Thanks, Brenda. So I guess just to answer the last most relevant section, I haven't sort of determined mapping using long reads across that breakpoint or those breakpoints. But these were similarly sized synthetic scaffolds between the two draft assemblies. So, we have several flavors of PacBio SQL2 read data. We've got the CLR and the HiFi. And BAMs for both, so I can go back and double check the inversion coordinates for that too. I don't know if that was everything for that.
Okay. Oh, Amanda is asking, isn't it the case that the inversion was found on both HiFi and CLR assemblies?
Yeah, it was within the grandis drafts. You know, and then with respect to dot plot, you know, with Thurberia also shows it. But yeah, so I guess that was how we were distinguishing whether or not. Yeah, we do. Yeah, Rob Waterhouse mentioned the high C data too. Yes, we do have the contact maps as well. And kind of looking at those plots, it doesn't seem like that the inversion is from a misassembly.
Okay.
Yeah.
That's cool. Yeah, I think
looking at the sequence.
I think Zach and I are both in agreement that we think it's real. So we're excited to see what it tells us as we do some more work on that.
Yeah.
I'm sorry to go back and forth, but I have one more question now that is for Perot. Chan Yu asks, were there a lot of repeat regions in the genome sequence of D. andersoni as seen in the genome of that blood feeding tick, which I'm gonna butcher the name, so I'm not gonna try.
Yeah, I'm familiar. So yeah, Exodes has a lot of that, and I haven't done a deep dive into the Andersoni genome yet, but you know what I actually suspect, and it's kind of relevant in that syntenic plot, is that I think a lot of that shared regions are actually just repeats or like kind of ambiguous sequences that are probably just repeat regions. So that's why they're mapping so much synteny between those two blocks. So anyways, there are, but I don't know if, you know, I don't have a quantifiable way to say it's just like Exodes or it isn't. But you do, there is a fair amount of repetitive regions, but you know, yeah, that's
Susan, do you have something to add? Okay. Well, I don't see any more questions for anyone. So I would like to just say thank you again to all our speakers. I think we would all agree that everyone did a fantastic job and we had some good discussion. If you have more questions that you want to ask speakers, you can always go to our Slack channel, which that was posted in the chat at the beginning and can also be found on the AGSx website to figure out how to get involved there. We also have a 4th session coming up in May. And so make sure you watch your email for links to that session as well. Brad, do you have any other things you would like to add before we end the session?
No, otherwise thanking you, Lindsay, for all your time and preparation work that went into this. Greatly appreciated. And also all the speakers as well. And I appreciate everybody taking the time to attend. And again, the videos will send out emails with the updated time or the time when the videos are posted on the YouTube channel for everybody to view and when the questions go into the Slack channel.
Okay, great. All right, thank you everyone. And hopefully I'll see all of you for the next session as well. So have a good afternoon.